Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs for the year 2014. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. Coming to June 2014 question paper. Isoelectric focusing experiments. Proteins are separated on the basis of their, they are separated on the basis of their mass to charge ratios. Answer is option 4. Then next one. Consider the structureless oligopeptide. They have given an oligopeptide. The number of bonds that will be cleaved by trypsin and chymotrypsin treatments separately are. So I have rewritten the oligopeptide over here. So they have asked for the number of bonds that will be cleaved by trypsin and chymotrypsin. Trypsin, as you all know, it cleaves on the carboxyl side of lysine and arginine residues. Whereas chymotrypsin, it cleaves the aromatic amino acids. That is, carboxyl side of aromatic amino acids will be cleaved by chymotrypsin. So here if you see, here you have got a K over here which will be cleaved by trypsin. Then you don't find any K or R. R is present at the ends. That will not be cleaved by trypsin because trypsin is an endopeptidase. It will not cleave at the ends. So only this K region will be cleaved by the trypsin. Then chymotrypsin has got two sides. That is, one is over here, that is next to between Y and G, and the other is between W and H. So chymotrypsin will cleave there. So you will have, they have asked for cleaved by trypsin and chymotrypsin treatment separately. So trypsin will cleave one and chymotrypsin will cleave at two places. So the answer here is option 1, 1 and 2. So coming to the next question, a 6.4 kb plasmid DNA has two restriction endonuclease sites, HIN3 and EcoR1. Complete double digestion of the plasmid with both the enzyme yielded two fragments, 3.1 and 3.3 kb. In order to study DNA repair process, a G:T mismatch was introduced in one strand of HIN3 site, and the damaged plasmid was incubated in a reconstituted repair system containing all the factors and enzymes required for repair. If the efficiency of the repair system is 50%, which one of the following band patterns on agarose gel will be obtained after treating the repaired plasmid with HIN3 and EcoR1? So you have got a 6.4 kb plasmid. It has got two restriction sites, HIN3 and EcoR1. If you digest the plasmid with HIN3 and EcoR1, you will get two fragments, that is 3.1 and 3.3 kb. Now you decided to study DNA repair process. So you introduced a G::T mismatch, and this was introduced into which site? HIN3 site. So the HIN3 site got damaged. And what you did, you, after damaging the HIN3 site, you incubated it in excuse me, you incubated it into mismatch repair system containing all the factors and the enzymes required for repair. But this mismatch repair system has only an efficiency of 50%. So which of the following band patterns on the agarose gel will be obtained after treating the repaired plasmid with both HIN3 and EcoR1? So first we will see what will be obtained upon complete digestion with HIN3 and EcoR1. You will get two bands, that is 3.1 and 3.3 kb. That is shown in option 2. But that is not the answer. So you can eliminate option 2. Now if you have introduced the mismatch into the HIN3 site, what all bands you will get? Now HIN3 site, once you introduce the mismatch, it will not get digested with the restriction enzyme because the restriction site has been damaged. So you will get only one band, that is 6.4 kb band representing the mutated DNA. So you will get 6.4 kb, but you will not get the 3.3 kb band. Okay, you will get only 6.4 kb. It will result only in a single digestion. So you will get a 6.4 kb band. And this plasmid was added to a mismatch repair system, providing all the factors and enzymes required for the repair. And this efficiency of the repair system is only 50%. So that only half of the plasmid molecules will become normal with 2 functional restriction sites, and the remaining half of the plasmid will remain mutant itself. So the mutant will give a single band of 6.4 kb. As you can see in option 3 and 4, you will get a band of 6.4 kb each. The mutant will give only a single band corresponding to 6.2 kb. 6.4 kb, while normal will provide 2 bands of 3.3 kb and 3.1 kb. So you will get 3 bands in an agarose gel. Now we can see that options 3 and 4 are correct, whereas options 1 and 2 are not correct. So out of 3 and 4, which is more correct? Now, fragments of 3.1 and 3.3 kb will always be equal. Whereas in option 3, if you see 3.3 kb will have more intensity than 3.1 kb band, which is not correct. The 3.3 kb and 3.1 kb band, they will have almost equal intensity. So option 4 is more correct. So the answer here is option 4. Then next one. A series of cell lines was created by fusing mouse and human somatic cells. In mouse human somatic cell hybrid, human chromosome tend to get lost before becoming a stable cell line. Some hybrid cell lines may carry human chromosome deletions. Each cell line was examined for the presence of chromosome and the production of an enzyme. The following results are obtained. So from the table you can see that the gene products A, B and D are formed in the cell line, and now you have to choose for a series for the chromosomal segment that is positive for all these 3 regions. So A is positive. If you see 1P and 1Q, A is positive but B is negative. So they are eliminated. If you see 2P and 2Q, A is negative but A is positive, you get the gene product. So they are not the chromosomal segments. In option 3, you can see that it is 3P and 3Q. But but the gene products are not formed for B. So they are not correct. 4P and 4Q also similarly you can eliminate. But if you see 5P, you can see that for A there is a gene product, for B also gene product, C or D also gene product, C and E doesn't have the product. So option the chromosome, which segment of the chromosome has the gene encoding for the enzyme? So it is 5P. So answer here is option 2. Then next one. Following are certain statements regarding the use of agrobacterium in plant transformation. So these are the statements they have given, and they have asked which of the following combination of above statement is correct. So agrobacterium tumefaciens causes crown gall disease and agrobacterium rhizogenes causes hairy root disease. That is correct. So whichever option doesn't have A, you can just eliminate. So 2 and 4 can be eliminated. Region A in Ti plasmid is responsible for replication. It is ORI region that is responsible for replication. So B is not correct. A is correct. Region D in Ti plasmid is responsible for virulence. That is also correct. Oncogenic region in tDNA is responsible for unusual amino acid synthesis. That is also not correct. So which and all are correct? A and C are correct. So your answer is option 3, A and C. Then next one. In transgenic mice line, LOX-P sites are introduced in the target gene A in the following manner. So this is the gene on either side of exon 2 you have got the LOX-P sites being introduced. The transgenic mice line was mated with another transgenic mice line where Cre recombinase is expressed only in B cells. So this particular transgenic line which is having LOX-P site was mated with another transgenic mice line which is having Cre recombinase. What will be the expression profile of gene A in the Cre-LOX recombinant mice? So you all know what is the Cre-LOX repair system. It is a technology that is used to introduce site-specific recombination events, and it consists of two components, that is the Cre recombinase and the LOX-P site. Now if you insert LOX-P site at deliberate locations in a DNA sequence, it will allow for very specific manipulations such as inversions can be obtained if the LOX-P sites are on the same DNA strand and are in opposite orientations. Deletions if the LOX-P sites face in the same direction. Okay, so here the LOX-P sites are facing the same direction. So the region exon 2 will be deleted. So we will see what are the statements. Gene will not be expressed in B cells as orientation of exon 1 will be inverted by Cre. Exon 1 will not be inverted by Cre because it is not flanked by the LOX-P sites. The next option, gene will not be expressed in B cells as exon 2 will be deleted by Cre. Exon 2 will be deleted by Cre because the LOX-P sites are in the same orientation. So option 2 is correct. Gene will only be expressed in B cells of the recombinant mice where the Cre removes the two LOX-P sites. That is also not correct. Gene will not be expressed in B cells as orientation of exon 2 will be inverted. That is also not correct. It will be deleted. It is not an inversion, it is deletion. If it has to get inverted, then the LOX-P site should be in opposite orientation. But here it is in the same orientation. So the LOX in the same direction. So yeah. So they will be deleted by the Cre recombinase. Then next one. A small fraction of clear cellular lysate was run on an isoelectric focusing gel to purify a particular protein which showed a number of sharp bands corresponding to different pi values. The protein of interest has a pi of 5.2. Therefore, the band corresponding to pi 5.2 was cut, eluted with appropriate buffer and subjected to SDS page which showed three distinct bands. Which one of the following inferences cannot be drawn from the above observation? Now first observation was several different proteins having same pi may be present at the single band on the isoelectric focusing gel. That is correct. So they have asked for which of the following inferences cannot be drawn. So from this first option, that particular inference could be drawn because in an IEF gel it is based on the separation is based on the pi values, and at that particular pi value you could get many proteins. So several different proteins having same pi may be present at the single band on an IEF gel. That is 100% correct. The next one. SDS page showed three distinct bands which may represent molecular masses of different proteins. Now once you elute out the 5.2 kb band corresponding to pi 5.2, then you subject it to SDS page, you will get three distinct bands, and these three distinct bands may be different proteins. The representing maybe having different molecular masses. So option 2 is also correct. The protein of interest may be composed of 3 subunits. That is also correct. It may be because the protein is having 3 different subunits at that particular PI. That is not at that particular PI. This particular protein at this particular PI is having is composed of 3 different subunits. That is why you got 3 distinct bands on an SDS page. So that is option is also correct. As the IEF gel showed a distinct band corresponding to PI 5.2, which is the PI of the protein of interest, the protein is composed of a single subunit. That is not correct. This PI 5.2 corresponding to that particular band, it could have several different proteins having the same PI also. So you cannot say that it is the PI of the protein of interest. There are so many proteins, several proteins of PI 5.2 which will show the band at PI 5.2 in the IEF gel. The protein is composed of a single subunit. You cannot definitely say the protein is composed of a single subunit. You got 3 distinct bands on an SDS page gel. So answer is option 4. The above diagram represents a 2 kb insert successfully introduced in between 2 BAMH1 sites of a 3.8 kb vector in desired orientation. So this is the 2 kb insert between 2 BAMH1 sites, BAMH1 sites of a 3.8 kb vector. So the vector is 3.8 kb in size. Into that you have introduced a 2 kb gene of interest. The HIN3 site on the insert and the EcoR1 site on the vector is also indicated. So the gene of interest is having another restriction enzyme site within it, that is the HIN3 site, and this is the EcoR1 site of the vector. If the insert was introduced in the opposite orientation, now if the insert was introduced in the opposite orientation, that is if it was first in the first condition, that is the insertion of the 2 kb fragment, it is from 5' to 3', but in the next condition it was inserted in the opposite orientation, means 3' to 5'. Which of the following statement is incorrect? Now if the vector if the insert is introduced in the opposite orientation, you will get the vector map like this. This is the 3.8 kb vector map. Here you have got 0.9 kb and here it will be 1.2 kb instead of 0.8 kb, and here it will be 0.8 kb instead of 1.2 kb in the original vector map. See here it is 0.8 kb and 1.2 kb. So this is the real orientation. Now if it was introduced in the opposite orientation, you will get the 1.2 kb fragment here and 0.8 kb fragment here. Now we will read the options. Digestion with EcoR1 will linearize the 5.8 kb plasmid. So 3.8 plus 2 kb you will get 5.8 kb. So once you digest the plasmid with EcoR1, it will linearize and you will get a 5.8 kb fragment. So that is correct. They have asked for the statement which is incorrect. Then digestion with BAMH1 will yield one 3.8 and one 2 kb fragment. So you are digesting with BAMH1. So it will cut over here and it will cut over here. You will get a 2 kb fragment and a 3.8 kb vector backbone. So that is also correct. Then double digestion with EcoR1 and HIN3 will produce 1.7 kb and 4.1 kb fragments. So with HIN3 and EcoR1 you are digesting. So this is in the case where the the gene of interest is in the right orientation, not in the opposite orientation. This condition, okay, I will tell you how. This is the EcoR1 site, this is the HIN3 site. So 0.9 plus 0.8, you will get nearly 1.7. kb band, and the rest will be 4.1 kb fragment. That is if it is in the right orientation. Now if you digest it with echo R1 and HIN3, if the fragment is inserted in the opposite orientation, then you will get 0.9 plus 1.2. That will be 2.0 0.9 plus 1. Yes, HIN3 and echo are 1, right? So it will be 0.9 plus 1.2 kb band, and A3, that will be around 2.1 kb. And a 0.8 plus 2.9 kb, that will be around 3.7 kb band you will get if it is in the reverse orientation. Now they have asked if the insert was introduced in the opposite orientation, which of the following is incorrect? So if it was in the opposite orientation, you could have got a 2.1 kb band and a 3.7 kb band. So that is in the case of opposite orientation. Now this one, the third option, they have mentioned it for that double digestion, they have carried it for the gene of interest being oriented in the correct direction, not in the opposite direction. And as per the question, they have asked for the incorrect statement. So answer is option 3.